Okay, so hi everyone, thank you for joining us. Today we welcome you to a CE3C conference, and we are pleased to have as our invited speaker Stephan Peischl, who I thank in advance for being available to share with us his work. The conference will be recorded, so if anyone wants to see it later, it will be available on our YouTube channel. Stephan will be presented by CE3C member Vitor Souza, to whom I now give the floor. Thank you, Vitor, and thank you also, Stephan.
So thank you. It's my great pleasure to host and introduce you Stephan Peischl. He was a colleague of mine, so we were both postdocs at a time at University of Bern, and we had really interesting discussions. And since then, I've been willing to have and invite Stephan to come to Lisbon. Unfortunately, the situation is still not optimal to have in-person meetings. So in any case, I'm very happy that we can listen to Stephan's work today. So just to give you an overview of the scientific path of Stephan. So he did a PhD at University of Vienna, supervised by Reinhard Berger, sorry. And there he worked a lot on very interesting and detailed methods.
Sorry,
Is someone talking? Maybe I cannot mute people. So yeah, so I was just saying that he did a PhD with theory and very important theoretical developments about the evolution of dominance in when you have, especially how does dominance evolve when you have migration and so on. So then in 2010, he moved to do a postdoc at University of Austin with Mark Kirkpatrick, where he worked on theory for evolutionary rescue and theory about the role of inversions in adaptation and population divergence. And then at the end of 2011, he moved to the University of Bern to be a postdoc with Laurent Escoffier. And there he did also very interesting and important work on range expansion. So when species have range expansions and increase their distribution area, and that's common in many species after glaciations that they had range expansions. And he did work to understand what's the genomic consequences of those range expansions, and in particular on the accumulation of mutations that can be harmful, so-called expansion load. And since 2015, he became a research assistant at the Interfaculty Bioinformatics Unit at the University of Bern. So he moved to a different floor of the building. And since then, he's been working on spatial models, recombination, trying to understand what happens to populations that are spread in space. Okay, and I'm very happy to have Stephan here, and thanks for giving this talk. Looking forward to hear it. Thanks.
So, thank you, Patricia, and thanks, Vitor, for the invitation and for this very kind introduction. So, as Vitor said, by training, I'm a theoretician. Yes, but I've tried to, since my early training, work on all kinds of like on data analysis, experimental evolution, and all these things. So I'm trying to connect theory and data. That being said, this talk today, titled Evolutionary Dynamics in Regions of Low Recombination, I'll do it the other way around. I will first talk about some curious observation that we saw in genomic data, and then we tried to see what kind of models are needed to explain this. So I'm using maybe theory to try to pose new questions. And this talk will be like 2 chapters that combine pretty much all of my interests, so population genetics and spatial models, but also the effect of recombination. And so whenever I'm not really clear or I'm using words that you don't understand, please don't hesitate to interrupt me at any time. Okay, so the first thing is it's called Untitled Transition from Purifying to Balancing Selection in Regions of Low Recombination. So this is joint work with Kim Gilbert, who was a postdoc here at in Bern. So Fanny Pouillet was also postdoc, and Laurent Excoffier was my old postdoc advisor, with whom I still collaborate quite a lot. And so here we're interested in genetic diversity. So this is one of the core goals of population genetics, to try to understand what determines genetic diversity, because diversity is what evolution acts on. If there's no diversity, then nothing can evolve. And usually, like, more diversity is better, because, well, diversity is the potential for the raw material for adaptation. It also prevents inbreeding, depression, and other things. And it's also in the light of species conservation. Usually we try to maximize genetic diversity. And so population genetics asks, what determines genetic variation? So this can be across a genome or across individuals, populations, different species. So what are the driving factors? And usually we consider two broad categories: natural selection, like any force where one individual is more fit than the others, or a mutation leads to a fitness increase and therefore establishes, or genetic drift. So these are all kinds of random forces: population size changes, like population contractions and expansions, and so on and so forth. And one key observation, so I'm going to focus in this talk about the first aspect: what determines genetic variation across the genome. And one observation that is now quite almost canonically accepted is that genetic diversity is correlated with recombination rates. So here, and these are a textbook, is a picture from a textbook where we have synonymous diversity, so diversity at presumably neutral sites, or sites where mutations don't have a strong fitness impact. And then we plot this against the local recombination rate. And we see that the diversity in regions of low recombination rates, we have little diversity, and it kind of increases as we move further away from this. And the reason for this is, so here on the left side in this panel, we see this typical valley of diversity that we see along a genome. And here in this case, we assume that there was a site under selection in the center, and this, when this mutation gets fixed, then it kind of reduces diversity at linked close by sites. And this effect is stronger for if the recombination is low, and weaker if recombination is large, and this creates this pattern. So this would be an example of a selective sweep. Here a couple of other examples of mechanisms that create this correlation of recombination rate and diversity across the genome. So the classic selective sweep is when a mutation, here this red one, increases in frequency, goes to fixation, and then it kind of leads to this dip in diversity because, well, where the mutation is fixed, there's no diversity, and then at linked sites we have the same pattern. If there are recurrent selective sweeps, so there's lots and lots of selective sweeps going on, then we see a similar pattern, but we see this interference pattern. So we see diversity is reduced in a more irregular pattern across the genome. And then there's a third kind of selection. So this is positive selection, where one new mutation is favored in the population. But if you have negative selection, so the current constant influx of deleterious mutation that get purged from the population, this also leads to a decrease in diversity, but it's not as pronounced locally and also a little bit more irregular. But in all of these cases, selection creates a reduction in diversity, and this is stronger in regions of low recombination as compared to regions where there's more recombination. This pattern is, so on this plot here, you see from a review paper, tau is the correlation coefficient between recombination rate and diversity. So if this is positive, then we see a positive relationship between these two quantities, recombination rate and diversity. And you can see that this is true across a large variety of organisms. Here in these woody plants, it doesn't seem to be the case for some reasons, but in many other organisms, we see this very strong positive correlation between the recombination rate and diversity. So this is a quite common and broadly observed phenomenon. And then this is the study that kind of made us think about what I'm going to tell you in a minute. So Fanny Pouillet, when she was a postdoc here, together with Laurent and Simon Eschbacher and Alexandre Thierry, had a study where they looked at how much of our genome is actually affected by background selection and gene conversion. biased gene conversion. And I found that like a large proportion of the genome is affected by this. And they looked at a measure of diversity. So they called it Duffy, which is the derived allele frequency per individual. So it's relatively straightforward to calculate, but this doesn't really depend. It's not in any given sample of different populations. So here you see this for 5 human populations plotted. It doesn't depend on the differential demographic history. And you can nicely see that if you plot Duffy, this measure of diversity against recombination rate in the human genome, for all these 5 populations that I'm showing you here, the pattern is almost exactly the same. So this is really a sign that even though like Africans, Europeans, East Asians, and so on had different demographic histories, this measure of diversity is very robust. Okay, so that's cool, and that was one of the interesting findings of their study. But there's something else. So we see that diversity decreases with recombination rate, but then for the lowest category, it seems to increase again, and does so in all of these samples that they looked at in the same way. And so this is maybe a small but a quite robust statistical signal. And this was something that was interesting because in humans you have very detailed information on local recombination rate, so you can look at this. You can actually look at not only very broad differences between like different categories, large, middle, and low recombination rate, but you can look at it in a relatively fine grid. And there we saw this uptake for the lowest recombination rate classes. And this is something we thought, okay, what's causing this? This doesn't seem to be just a statistical artifact, so we wanted to look at this. Then we went to the literature, and there is, of course, Brian Charlesworth is also working on this. He looked at this from Drosophila and found similar patterns there. And they studied an old idea that has been called associative overdominance. That's nothing else than just saying that under certain conditions, selection against deleterious mutations can lead to an increase in diversity. So here they plot the relative diversity. So 1 would be what you expect in the neutral case. And then they show this for one particular case, how over time diversity can increase at linked neutral sites. So essentially this is a potential explanation. The process that leads to this is relatively complex, so I don't want to go into detail, but I just want to say that this purging of if you have recessive deleterious mutation, so you purge the homozygotes, that leads to an increase in heterozygosity, and that can spill over to neutral sites. But also note that this is a relatively modest effect. So we're saying here on the order of 1% increase. And this is only true. So this is only happening for relatively specific conditions. So this could be a potential explanation, but this is clearly not strong enough to explain what we see. And yes, and so in this quite impressive and really good work by Li Zhao and Brian Charlesworth is assuming a 1 locus model. So we look at one particular site where we have deleterious mutations. So we wanted to extend this model to a 2 locus and then multi-locus model. So what does this mean? We have 2 loci, locus A and locus B. Each locus has 2 alleles. And then we can have all of these 9 combinations of 2 locus genotypes. And here I just write down the fitness values of all these possible combinations. And the assumption that we make is that mutations are recessive, so selection acts only against homozygotes. So here we have the homozygotes, they each have a selection coefficient that reduces the fitness, and then here's the combined effect of these 2. And there is a recombination rate between these 2 loci, and mutations occur at each locus at rate U, and the capital U is the total deleterious mutation rate. So the simplest possible model. We extended this to partially recessive mutations, but I'll just keep this model for now because the results don't change much. And here is what we observed, or this was also like, this is the what we were looking for. So here on the left side, you see the case for strong recombination, and here on the right side, you see the case for no recombination. So each line is a haplotype frequency. So we have the wild type, 1 deleterious mutation, 2 deleterious mutations, or both. And then we see different dynamics over time when we have strong or no recombination. So what we see here is in the left, we see this mutation selection balance, where the wild type, the most fittest haplotype, remains in the population, and mutations are at some given frequency. and we have relatively low levels of heterozygosity or whatever, however we want to measure diversity. But if there's no recombination, all other parameters are the same, then we suddenly observe that the wild type gets lost from the population, so it approaches 0. And then the other two haplotypes, AB and big A, small b, and small a, big B, are maintained at a frequency of 50%. So this is maximum diversity, essentially. And so why is this? The reason is, this is, and again, this is the old idea. This is so-called pseudo-overdominance. This has been studied by Ota and Kimura in 1969. And essentially when the zero mutation haplotype, so the ones AA and BB without mutations, get lost from the population for some reason, then suddenly we have a different fitness scheme. We have balancing selection that favors the heterozygotes, so maintaining the heterozygotes at a very high frequency. So this is an interesting phenomenon, but with a two-locus model, this is extremely unlikely to happen if you use realistic parameter values, because rarely ever will this wild type become lost. If we really just look at this toy model. So therefore we try to extend this model to an arbitrary number of loci. And what we try to predict is the equilibrium frequency of this zero mutation haplotype. And it turns out we could calculate it or approximate it as a relatively simple formula. And I will tell you in a minute why we want to do this. So N is the number of loci that we have. And mu is the scaled mutation rate, and rho is the scaled recombination rate. So we measure mutation rate and recombination rate relative to the strength of selection. So and what follows from this model is that if this P0, the frequency of the zero mutation haplotype, approaches 0, as we see here in the right case without recombination, then we will maintain this haplotype frequency, or these haplotypes at the frequency of, depending on how many haplotypes will be present at the equilibrium, or something on by 1/n. So there will be a transition from this purifying selection that always purges these mutations to balancing selection that maintains these haplotypes. And the reason is simply because you want to increase heterozygosity, because you cannot get the zero mutation homozygotes, so you want to have as many heterozygotes as possible. And this is this transition from purifying to background selection. Okay, and so if we now can predict this P0 this is a purely deterministic model, we can also ask when does it approach 0, and so when do we see this transition from strong recombination or from purifying to balancing selection. And here's now these results from the multi-locus model. So the key goal here was to see if this can occur for reasonable parameters. And then there's an interesting thing that we see. So in the leftmost panel here, you see this frequency of this P0. So if this approaches 0, then we should see this balancing selection. And so we can see, first of all, it's not only true for like a recombination rate of 0. We see here, if the mutation rate is high enough, then we have a relatively broad range of recombination rates for which we see loss of this zero mutation haplotype. So this again for a two-locus model, the lines are the analytical model, the dots are simulations. But more interesting is if we now look at a multi-locus model with N loci. So in this middle panel here, where we plot this P0 as a function of the number of deleterious loci in our model, and we keep the total mutation rate constant. So mu is the mutation rate, and these are now relatively realistic parameters. So we have a quite small mutation rate. So this is the ratio of selection intensity and mutation rate. So mutation rate divided by selection intensity. And here we have relatively low such values. But if we have just a couple of dozens of loci, we can lose this zero mutation haplotype and observe this transition to balancing selection. It's also interesting that this happens quite sharply. Like really at some point when we reach a certain number of loci, then the frequency goes down almost like very, very abruptly. And if you plot this now in the original parameters, just in panel C here, it's a function of the recombination rate and the selection coefficient. The important thing here is that blue is the region where we expect the loss of this zero mutation haplotype and balancing selection. And we can see that this is most likely for intermediate selection coefficient, simply because if selection is too strong, then mutations cannot increase in frequency by drift. And if recombination or selection is too weak, then recombination is so strong that it constantly reintroduces the fittest haplotypes, and we can never get the loss of the zero mutation haplotype. Okay, so this the point here of this exercise is to see that, well, for realistic parameters with just a couple of deleterious loci, we should observe such a transition to balancing selection. And this is where then Kim Gilbert, one of my favorite collaborators these days, came in and said, okay, let's try to simulate this with SLIM. Let's try to come up with a simulation framework where we have like a mutation rate of 1.45 times 10 to the -8. So this is chosen from, this is a human, this is in the range of what is considered realistic for humans. We used a relatively simple recombination map with just 6 chromosomes per individuals and constant recombination rates per each chromosome, but varied those. We had 10,000 diploid individuals, constant population size, no complex demography, and then varied the strength of selection, the dominance parameter age, the recombination rate, the proportion of mutations that are neutral versus deleterious. So all of this was kind of tailored to be a simplistic version of what we expect in humans. And here is our sanity check. So these are codominant mutations. So this means the individual effects of mutations just add up. And this is the case where we would expect only background selection to reduce diversity. And that's what we see. So the gray line here, this baseline that we see, is the diversity we expect in the neutral case. So in here we have nucleotide diversity traditionally measured by pi. So this is the average number of pairwise differences between sequences. And here's just the average derived allele frequency. And in both cases, we see a reduction in diversity if recombination rate is low. And this is most strongest for intermediate selection coefficients. So this perfectly fits theory. This is just to check that our simulations do what they are supposed to do. And now let's have a look at fully recessive mutations. So here we have quite different pattern. The important thing I want to highlight here is that in both cases, so again, the gray line is the neutral expectation here and here. And again, for intermediate selection coefficient, depending, of course, on other parameters, we see this increase mostly in diversity, depending on how we measure it, but mostly for sites in this area of 10 to the power of -5 to 10 to the power of -3. So relatively weakly deleterious mutation or deleterious mutations of intermediate effect size. And the other thing I want to highlight is that, so when you look at nucleotide diversity, this is a threefold increase in diversity in the most extreme case. So that's a massive increase in diversity. So that really suggests that this can no longer be explained by pure dissociative overdominance. Here must be something else going on. And if we look a little bit closer, then we see it also matches this prediction from theory that in the regions of low recombination, we see this effect most strongly for intermediate selection coefficients. So here for the red lines, and in some cases the orange, and turquoise line, but never for the very strong selection coefficient. Sorry, I couldn't understand this. It was very chopped up.
Yeah, I don't know if it was a question or if it was someone with a microphone issue.
Okay. So, okay, but this is a very complex pattern that we see here. The important, or the key things is that it also happens for intermediate dominance. So this is H equals 0.25 means that we are somewhere right in the middle between codominant and fully recessive mutations. It's of course very strong for recessive mutations, and we see it in both measures of either diversity measured as pi or by the derived allele frequency. And yes, I'm not going to show this here, but we also looked at the haplotype structure and all these things. And we really see that we get this clusters of haplotypes present at average frequency. So it really looks like it's hard to make a clear decision whether it's this pseudo-overdominance or not, but the data clearly looks like this and also matches all the predictions from theory. And then we went on to see if we can find such regions that show this signature in human genomes. And for this, we looked at our two, so we realized that these two measures, nucleotide diversity and Duffy, capture different aspects of diversity. So and here in this plot, we calculate the increase in Duffy, or the difference in Duffy between low and medium recombination regions. So that means, and here the same, but for pi. So in lower values mean that, or if you here in the left, this means lower mean allele frequencies in low recombination regions, and you have higher mean allele frequencies in low recombination regions, so more diversity in low recombination regions as compared to high recombination regions. And here we have the same, you can roughly say, here we have fewer polymorphic sites in low recombination regions, and here we have more polymorphic sites in low recombination regions. So what we then see in this plot, and so we plotted this for all parameter values for different dominance patterns and different selection coefficients. And what you can see is that for either partially recessive mutations or very strong selection coefficients, we are in the region where background selection reduces diversity in most cases, and we see the strongest effects for large S. So this is, again, makes a lot of sense. And here, this region is where we see an increase in diversity in one way or the other. I would say that the most interesting part is this one here, because these are the most realistic parameter combinations. So these are these are relatively strong selection coefficients here, and also it's complete recessivity. So here is where we all find the intermediate recessivity and the intermediate selection coefficient. So this is really the ones that are realistic, more realistic parameters. So and then we wanted to look in human populations, and this was mostly the work of Fanny Pouillet. So she calculated these statistics for 10 different human populations from the 1000 Genomes Project, and tried to do a scan for regions where we see excessively large increase in diversity in both measures of diversity. And here's an example where we scan for these outliers, and you can see that the shape is kind of similar-ish, except that we don't see these extreme parameters. So this is for the Yoruban sub-Saharan African population, and we made very strict criteria. So we said that this excess needs to be present in all 10 human populations and in both statistics. So and the reason for this is that if it's really some form of balancing selection that maintains this diversity, then it should be relatively old polymorphism, and therefore we expect to see it in all populations. Also, we want to be just on the safe side and try to find a robust statistical signal. And so overall, she identified 35 candidate regions using this relatively strict criteria. And as a sanity check, we also picked up one of those 35 candidate regions, and I'm just showing 3 examples here, was the MHC locus, which is a very known example of balancing selection. And clearly, since we're looking for signals of balancing selection in regions of low recombination, this signal, this region should pop up, and it was actually one of the #1 candidates. And here in this small plots on the right, this is a little bit hard to read, but this is essentially a normalized side frequency spectrum. It shows the derived allele frequency that we find in these candidate regions for all, it's just a distribution of this. So this means, and if this line is flat, so we normalize it to the flat line, means everything looks as it should, like everything looks like in a neutral population. And any deviations from a straight line at 1 are show an excess of mutations at a given frequency. And this is very hand-wavy, and I wouldn't put my head on fire for what I'm saying right now, but I still think it's interesting. What we see in all these cases is that we see these spikes. So the blue lines are the low recombination regions, and the orange lines are the high recombination regions in these candidate regions. And in these examples we often see these spikes at intermediate frequencies around 50%. So this is really also indicative of this maintenance of complementary haplotypes here. because we have multiple mutations that segregate at the same frequency, at the same very high intermediate frequency. And interestingly also, and again, this is very, very hand-wavy, but in this MHC locus where we know this is classical balancing selection and not this maintenance of complementary haplotypes, it looks more like it's a shift towards high frequency variants. So it means some variants at the high frequency, others are at low frequency, whereas here we have multiple sites that seem to be at intermediate frequency. We also checked for what kind of genes do we find in these regions, and we find these are not like pseudogenes or completely neutral, and there are functionally important genes found in these regions. But with this small number of regions, the small number of sites that we had, there was no way to do like proper statistical analysis of this. But it's still something that I think was interesting to look into. And of course, when we remove these 35 candidate regions, then of course the signal, the statistical signal disappears. And then we have a so this was the opening figure that I showed, but I don't want to, again, this feels like circular reasoning again. That's what people often ask. Does this increase in low recombination region diversity then disappear? Of course, it disappears because we also just selected the most diverse regions. So that would be circular reasoning. And yeah, so with this, I would like to wrap up this first part. And so in this first part, I've shown you that selection against recessive deleterious mutations can increase diversity at linked sites in regions of low recombination. I think this is really important because it's a potential confounding factor in scans, in any kind of genome scans, right? Because something that looks like what we believe is balancing selection, so really it's positive selection maintaining diversity, but it's actually just reducing the harmful effects of deleterious mutations. And then I guess Vitor can tell you much more on this. Should also be interfere with scans for local adaptation when you have like more spatial models. And it's definitely something that we want to keep in mind when we do these genome scans. And Brian Charlesworth studied this in Drosophila where they found similar patterns, although not as strong as in humans. And so there's if you're interested in this, Simon Charlesworth is a very good theoretical paper, and then Peischl and Charlesworth is a paper where they look at this in Drosophila. A similar process has also been studied recently by Bern et al. So this is a collaboration with Claudia Banks' lab, also here in Bern, where they looked at this kind of crystallization process in inversions. And if you're interested in the paper, here's the reference. It's just appeared last year. Yes, and currently we're trying to extend this to other scenarios, including selfing and so on and so forth. Just check. So, yes. This was the first part where I think we could, or like where we found these old ideas, from the 70s, going back to the 60s and 70s. I think that they're still relevant in a genomic setting where they can actually be really important. And the second part I want to talk about is now more of a spatial model where we talk about gene surfing. So I'll explain what this is. And now instead of looking at this overdominance or this selection for hybrids, I'm going to look at selection against heterozygotes or hybrids. And this is a collaboration with Antoine Monet. It's a postdoc in my lab currently, and again, Kim Gilbert from the Institute of Plant Sciences. So this was motivated by ring species. So ring species this was this idea that when a population expands from original population along a geographic area, and then kind of keeps evolving as it moves, that when it meets again, so like think about some birds or plants moving, expanding their range across a lake or something, then when they meet again, they are incompatible because they have established so many incompatibility levels. So there have been a couple of examples, but two quite iconic ones were these salamanders at the coast of California that expanded around the Central Valley, or these greenish warblers at the Tibetan Plateau. And yes, and then at some point, Jerry Coyne, like famous scientist working on speciation, tweeted that there are no ring species and wrote a blog entry about this particular paper, which it's a very nice paper, looking again at now with more genomic data at these greenish warblers. And what they found, and this is what that so here in Siberia would be this secondary contact where we see and observe like strong reproductive isolation, but we also see partial reproductive isolation between neighboring populations in other regions. So here we have a very strong cline in SNPs, that there's no apparent environmental conditions that drive this very strong cline. So it's not clear what's driving this, but there are multiple clusters of partially reproductively isolated neighboring populations, and not just as so the reproductive isolation is not just happening here, but also here, maybe here, different positions. And this kind of brought back, okay, so there's a range expansion like along some like mountain or lake or some obstacle. And this is really close to what I've studied a lot with here in Bern. So we want to look at this. What happens, do underdominant alleles during range expansions? How do they evolve? And we're really looking at the simplest model of selection against hybrids that can kind of create such strong clines. And this is one locus with two alleles, and heterozygotes are less fit than either of the two homozygotes. That's underdominance. So these alleles have a lower fitness, or these genotypes. And what I want to calculate is what's the probability that such mutation establishes during a range expansion. So here's our typical sketch of a range expansion. We have the population density on the y-axis, space on the x-axis, and this population colonizes space from left to right. And now I have here, I introduce, let's say, an underdominant mutation is present here at a low frequency here in red. And what can happen is that it kind of surfs this expansion wave and colonizes new habitats just because it's lucky to be on the expansion front. It causes a large part of the species range. And then this can look like this. So here I plot now the frequency of this allele as a function of space. This would be the classical example of a hybrid or tension zone, where the big A allele is favored, where in the region where the big A allele is more abundant than the small one, and vice versa. And therefore we get this stable cline in allele frequency. So this was been studied by Nick Barton extensively in the 70s and 80s. So that's the classical model for these hybrid or tension zones. And now, how can we study this in the context of a range expansion? Well, usually, so here we have this range expansion is more continuous one to the left. But I'm going to use this model to the right, where we discretize space into demes, and then just assume that we have a number of F founders colonize a new deme, and then this deme grows for T generations until they reach carrying capacity, and then we have the same process again. So essentially, we disentangle gene flow and growth here. It's a simplification. And I see that I'm maybe need to be a little bit quicker. So the key point is we can calculate the probability that such a mutation, an underdominant mutation, takes over, surfs on this wavefront using classical diffusion theory. And the important part is we find that the strength of selection is reduced by 1/3 relative to classical deleterious mutations, and the amount of drift is increased by n divided by f. So this is the ratio of total number of individuals per team divided by the founders. So that means if we have a small number of founders, then we have lots of drift, and if we have a large number of founders, then we have less drift. But for reasonable combinations of these parameters, we see here the fixation probability of function as a function of the initial frequency. And the more sigmoid this is, the stronger selection it is, and the less likely it is that surfing will occur. But the more we have a straight line in here, the more does this mutation behave as a neutral mutation, and the fixation probability is just the initial frequency. And we see that in relatively quickly expanding populations, this is when the time t between the colonization of two consecutive teams is relatively small, or if there's a small number of founders, surfing of these underdominant mutations seems to be quite likely. And then Kim again did individual-based simulations of this surfing, and to really show that this also happens in more realistic scenarios. So she did a 1D landscape of 150 teams. She introduced mutations in a particular time point so that we can easily track them. Heterozygote fitness was pretty low, so 0.9. So this is a 10% reduction in heterozygotes, and a relatively high migration rate. So this is 5%, like a proportion of 5% of individuals moves one gene to the left, the other 5% to the right on average. And the carrying capacity is like 100 individuals per team. These simulations were performed in SLIM and are like more reflecting some continuous space model. And what you see here, this is a bit difficult to read, but I still think it's interesting. So I'm showing you now the allele frequency across time. So black lines mean at the point when this, or the color is the age of the allele. At time t = 0 is when the allele gets introduced. And then this bluish turquoise color is 10,000 generations later. And you see that this Cline establishes an irregular way in the beginning. And then instead of just random movement, there seem to be some quite consistent movement, and then it comes to stop and stays here at where the red line is. So this Cline first establishes, then moved a little bit to the back or drifted around, then moved to the front and then back again. So it kind of seemed quite unrandom to us. And then when we look at what happened at the second locus, so these were mutation close by locus, we see that this was introduced around here and then established a Cline very quickly and then just moved backwards. And then when they perfectly aligned, these two Clines, so were at the same spatial position, they stopped. And that would, yes, and that's something that seemed curious to us. Why would they do this? Because each Cline is, can move around just by drift as they want, but why do they seem to attract each other and align themselves in the same position? And then Antoine wrote down a continuous space to locus model. Just look at the fitness scheme, but essentially we have now two loci combined with recombination rate. These are diffusion parameter, our migration rate, and so on, some details. But here the idea is now that only heterozygotes are selected against. So it's the opposite fitness scheme as in the previous chapter. And yes, and there's no epistasis or nothing fancy going on. And what he found is that if you don't have, so now on the left, on the right side, you see the dashed lines the initial conditions, and this is just an animated GIF that goes through the same loop over again. But you see that these two Clines over 200 generations attract each other until they meet, and then they would just stay there. This is the case for no recombination. This is a numerical solution of this model. If we have strong recombination, same model, with strong recombination also for 200 generations, you see that the Clines kind of move a little bit towards each other, and then they move more slowly, but not really. So everything is the same except that recombination rate is different. So clearly what we see here is that recombination rate modulates this attraction of these Clines. And so, and this is why I say like revisiting old ideas. We then looked at the literature, and if you, everything is already in Barton 1989, a paper by Nick Barton. It's not, he didn't spell out the attraction of these Clines. He looked at already overlapping Clines and how the strength of selection changes for those. But yes, all the arguments are already in there. And what's happening is that, so if you have two Clines here, like a gray one, a gray allele, and the blue one, the blue allele, and they overlap. What we have is that, so there is, selection will create correlation between allele frequencies at both loci, because we select against heterozygotes, so we will have more homozygotes. So that means that the small a and the small b allele will be correlated, and the big A, B and the big small b allele will be correlated. And this, and so whenever we have selection for one of the alleles, like the gray allele, it will spill over to the other locus and vice versa. And the strength of this correlation is inversely proportional to the recombination rate. So this is, you can read this off the equations. What it means essentially is that under dominant mutations, so if I look at the right side of this line, this is where the gray allele has the higher frequency, like it's above 50%. And here the gray allele, this indicated by this line, is below 50%. So the left of this line, we have selection against the gray allele, because when we have underdominance, we have selection against the rare allele. And at the other, and then here we have selection for the gray. And if this spills over to the blue line, this means the blue line will be pushed up here to the right of this line and pushed down to the left of this line, which is the same as moving closer to the gray line. And then you can flip this argument and show that the gray Cline will also move towards the blue line. So this is really LD creates a correlation between allele frequencies, and therefore this LD will move these clines closer to each other. There's a super interesting preprint by Dennis Rose and Alvaro et al. From 2021. Very mathematical, but very interesting, where they studied this in more detail, not or slightly different aspects of this problem. And yes, and what Antoine and I then did, we wanted to see if we can actually calculate the speed at which these two clines move towards each other. And here we can calculate the speed and can even show that it's directly proportional to linkage disequilibrium, which is just the amount of correlation between allele frequencies at different loci. And this is all expressed in units of the width of this cline. So that's a relatively simple formula that we can even evaluate. And then we can compare the speed that we calculate against our numerical solutions. And the dashed line is the analytical prediction, and the solid lines are what we get from numerical solutions. And we can see that, so I'm sorry, I forgot to label this. The green one is for weak recombination, and the yellow one is for strong recombination. So we can really see that there is some kind of optimal distance between clines. So that would be one spatial unit, so which is given by the cline width, where attraction is fastest. Otherwise, if the clines are too far away from each other, the strength of attraction will be very, very low, and it will be very slow. Yes. So what this means, and now this is like what we can take home from it, is if we have spatially overlapping clines and recombination is not is sufficiently weak, then we can expect that two neutral hybrid zones that are not linked to any kind of environmental variation will tend to overlap and amplify each other. This is a preprint that we have just submitted and will be, I think it's already in press for especially in philosophical transactions. So if you are interested, you can look it up. And so to summarize this, it really shows that range expansions can actually facilitate the establishment of hybrid zones in the absence of any kind of environmental variation. And what I want to do next is see, so we have now in this purely deterministic case derived this strength of attraction or the speed. Next step would be to compare it to the strength of drift, because drift also moves clines around, and if the strength of attraction to the selection is stronger than the strength of drift, then we would expect, then we can somehow calculate the critical range of overlap that we need to have such that clines actually attract each other. We want to look at the molecular signatures of these spatially aligning clines, because that could really tell us, when you look at these greenish warblers, are there any signatures that can tell us, well, this is really due to clines that have moved, have aligned themselves spatially. And then the next thing would be to include local adaptation and extend asymmetric cases. And this has been discussed by Nicolas Bern in the 2011 paper. But here I want to sort of like, if there are locally adapted clines that essentially like really can they be pushed out of place by other clines, or will they all tend to be in the same place, and so on and so forth. So essentially, how do different incompatibilities or how do different pairs to reproductive isolation interact? Yes, and I think with this, I just want to thank my co-workers, collaborators on this, so especially Kim, Fanny, Laurent, and Antoine, and also my lab and the bioinformatics group, but also the CMBG lab with whom I collaborate a lot. Vitor, of course, because we discussed this a lot. Brian Charlesworth, who is also quite big help with the first part. Nicolas Bern, who has discussed the second part a lot with me, and also Nick Barton, who has discussed the second part quite a bit. And yes, if you have any questions, I'm very happy to take them. Yes, and maybe I'll just close with this slide here, which was something that we fought quite a lot over these projects.
Okay, thanks a lot, Stephan. Sorry.
Yeah, no, I was just saying thanks with Stephan, and we don't have questions in the chat, but I assume people want to ask you questions directly. So we have some time for discussion, so please.
Yeah, so maybe I can start. I think So very interesting about this Cline attraction. So I was just wondering, I have two questions. So the first one is, what is causing this underdominance? And I guess it's incompatibilities, but just, you don't include any epistatic interactions, right? So it's just one locus incompatibility, right?
Yes, yes.
Okay, so it's just something that it could be, and to connect with the ring species, that the two expanding waves accumulated two alleles that in a heterozygous state are incompatible. Or it could be, for instance, an inversion, where one of the inversions in heterozygotes will have a lower fitness. So something like this, right?
Absolutely, yes.
So the question would be, so what if you have a more classical incompatibility with two loci? Do you think you will still have this? Okay, but maybe this is more to discuss later. I see. But so in a way, what would be if you have epistatic interactions? And so do you think it could stop this if, okay, because here you're assuming it's multiple, right?
Yeah. So I think maybe I can answer the first one. So you would look at Dobzhansky-Muller incompatibilities or any kind of two locus incompatibilities, right?
Sorry, can you say that again?
So you were talking about Dobzhansky-Muller incompatibilities. Yes. So they will serve even more easily because they are neutral in a way. Only the second mutation would be selected. So I think it could be extended more or less straightforwardly, and I don't see any reason why conceptually it should be different.
Yeah. So my second question was the implications of the fact that the Clines get attracted in regions of flow recombination. So if I understood correctly, you said that in regions of flow recombination, it's where this attraction occurs more often. So could it be that when we look at real data, we tend to find these Clines? Could it be that we interpret sometimes as evidence of local adaptation of mutations that are adapted to the different environments, or it could be incompatibilities? But and the fact that they are coinciding in different regions, we usually try to find environmental variables that correlate with that.
Yes.
Say that it's local adaptation. And what you're saying is that, and we usually find that in regions of flow recombination. So does it mean that this mechanism can explain what people have interpreted as evidence of local adaptation?
Yes, yes. I mean, I wouldn't, I think it's one alternative explanation that could also make sense. But I think in reality, it will, if this happens in reality, I think it will also often, and that's, I think what is an interesting follow-up study, and what other people are also doing is, because if you have a Cline to the local adaptation, which, I mean, they clearly exist, this Cline would also attract neutral Clines, right? Because so it could be that I would rather say that locally adapted Clines, or range expansion, could set the stage for establishing these Clines that are neutral in the sense that they are not linked to any kind of environmental relation. They can be anywhere in space. They're the same everywhere. That they will maybe set the stage and then amplify or move to the region where you have some kind of environmental gradient.
Okay.
So I think it would really facilitate.
Yep.
Yeah.
Yeah. Okay. Thanks. So I see that Sarah has a question.
Yes.
Sarah, you're muted, maybe. Sarah, are you still there? Hello. Can I? No. Okay.
Hello. Hello.
Yes, we can hear you now. Now we cannot hear you.
Então és tu, é isso?
Fala lá.
Hello. Can you hear me?
Hello. Yes, I can hear you.
Yes,
Hello.
yes.
Okay. So thanks. I have actually two questions, and I'm sorry because they might be really stupid, both of them, but I didn't really have time to assimilate what you say and think about the consequences of it at the same time. So the first one is that I think I understood what you said in the second part, but I'm not sure how that relates to the occurrence or not of ring species. I don't think you made this connection, and I could not make it myself. And the second concerns the first part of the talk, where I was wondering what are the consequences of this finding for Muller's ratchets? Because I mean, I could not think about it again, but I think that I think basically now it goes both ways, right? So Muller's ratchet affects the evolutionary of recombination, but recombination also affects Muller's ratchet in another way, right? So I'm not sure. I mean, I just want you to tell me, because I could not think about the answer properly. Yes. Thanks.
So thank you, and I want to say that these are two fantastic questions, both very good. So yes, I think I was a bit misleading with the ring species. I don't want to explain the occurrence of ring species. I just said that this data that popped up that showed, oh, ring species are not what they seem to be, because there are incompatibilities or partial barriers or barriers to reproductive isolation, partial barriers all over the species range. Seems that something else must have been going on, and this made us think about, can these incompatibilities also establish, so this one locus incompatibility is established during a range expansion, because in single mixed populations they usually don't establish. So it was not clear what causes this pattern of multiple barriers to reproductive isolation in these ring species. And so,
right, Stephan, sorry to interrupt. So the argument that there are no ring species is because you'd find these Clines all over the place.
Yes.
And not really where they met.
Exactly. That was a bit what Sherry Coyne said. That,
so you're giving reasons. So you're saying that Sherry Coyne is right, basically, right?
That it makes sense. It makes sense that this could happen during range expansions. But I also agree that it's very difficult to test such a hypothesis. But I think it's an interesting thing to keep in mind. And yes, and maybe one can see if one can find like. We need some kind of molecular signals, right, that we can look for. But that's the idea. Yes, I'm saying that, yes, it might not be ring species in the classical sense, but there's something, but it's still interesting, right? That this range expansion can really lead to this establishment of incompatibilities at various positions in the species range without any apparent ecological boundaries.
Okay, okay. And then the first question?
The first question, yes, Muller's ratchet. So that's very good. I was a bit sloppy here because when we look at this, for instance, where do I have a figure? Let me just. So in this figure, I think you see my slides now. Oh, that was the wrong button. Sorry. So when we looked at Muller's, we essentially don't think about Muller's ratchet here, and the reason is that we looked mainly at parameter values where Muller's ratchet doesn't matter. So here, when we have various mutation rates, and this loss. So the interesting part is, so this loss of the zero mutation haplotype is not the same as in Muller's ratchet. Because in Muller's ratchet, the loss of the zero mutation haplotype is driven by like the mutation rate is too fast relative to purging.
Right.
But here it's more complex because these mutations are recessive, and if I have many loci and I have at each of these locus I have mutations at a relatively low frequency, all of them, they're essentially not visible to selection because they're recessive. And the more loci I have, the more heterozygotes I have, and these heterozygotes are all invisible to selection. That's our, because this is due to this fitness scheme. And therefore, with many loci and even a relatively small recombination rate, you can lose the zero mutation haplotype. I mean, you could call it Muller's, or you could call it a diploid.
No, sorry, that's not my point. My point is that you have now a reason why there is still diversity, even though even though you have a sort of a Muller's ratchet happening, right? Because you're purging
Yes.
So the pattern seems to me a bit like Multi-Locus, because the intermediate effect, you
Yes.
keep the others, you know, whatever. But now you have a way of keeping diversity. So basically there should be less selection for recombination due to Multi-Locus, and this might be an explanation why you don't really see Multi-Locus happening so much.
Okay.
I don't know, maybe, I don't know.
Yeah, that's in diploid organisms at least. Right?
Yeah.
That's very interesting, yes. And I think so it would be, that's something we didn't do, and now that you say it seems like something we should have done, is just to also to look at the mutation load, how it changes, right? Because this is this loss of the zero mutation haplotype is not necessarily an increase in the mutation burden. Because you mask it.
My point, exactly.
Very, very interesting. Thank you. Yes, I'm going to look into this.
Okay, cool. Thanks.
Thank you.
We still have one question from Evu.
Yes. Okay. So sorry. So I was wait on 1 second. Okay, Brian. So I was wondering on the first part, when you have this increase in diversity because of selection on associative overdominant loci, and I was trying to make a link to some of work on Nick Barton in the early 2000s, but for true overdominance loci, where they showed that actually it depended a lot on the level of epistasis and the type of epistasis that you would see, such that in some cases, although you would always have an increase in heterozygosity, the haplotypic diversity would depend a lot on whether you would have a negative epistasis or a positive epistasis. Mm-hmm. Extreme, if you had a lot of positive epistasis, you might end up with just 2 haplotypes at intermediate frequencies, and that would be all. Mm-hmm. In your model, actually, I don't think you included, you just assume additivity. But is it because you don't think it will actually matter in this case if you have epistasis, or is it just a first approach and then you will eventually may extend to that?
Yeah. Yes, it was definitely the latter. We didn't think about epistasis because already with partial dominance, so we already had partial recessivity in here, like where you have instead of these ones here, you have 1 minus HSs, and already that makes it much more complicated. So it was more that we aimed for the proof of principle here.
Okay, great, thank you.
But yes, epistasis, and that's also something in both things where we try to include epistasis or simple forms of epistasis and see how they change things.
Cool. Thank you.
Okay, I don't know if there are other questions, and in any case, it's already past the time.
Yeah.
So, are there any other questions? I guess maybe there's time for one more. No, Patricia, tell me. If
no. If there is one more question, yes, we can leave, or if not.
Okay, so there's one question here.
Okay.
I can see. Sorry, just a second.
Thanks. I have a question about the second part in second expansion. So because you have like this expansion, you also have a risk of activating like deleterious recessive mutation. And then how having a mutational loss a bit could create like some hybrid figures that may contract the Clines. Do you have any idea how it could affect like the result, if that makes sense?
Sorry, I didn't get the last part.
So basically, this idea is that you have this French expansion, so that of gene surfing, so you could also have like recessive deleterious mutation accumulating, right?
Mm-hmm. Yes.
So having this will generate some hybrid figure at the right position. And I just wonder how the two mechanisms may interact with it.
Yes. First, I mean, I think Saliotto's group had a recent paper on this.
Yeah, that's because the question comes from.
Yes, and we also looked at this in an earlier paper, but not in that much detail. We looked more at heterozygosity fitness correlations and how this would interact. Interesting question. I guess it will depend on, again, the recombination rate between these things. And I'll just, yes. I honestly don't know what I would expect. If I would have to make a guess, and I would say if you do some simulations in, let's say, Slim or so, and have a mixture of these things, I would think that you would find a mosaic of these different processes along the chromosome. That's my guess. But yes. Yeah.
Okay. Something to think about. Yes, I still think that many of these old ideas that have been here since the 60s and 70s, now we should reconsider them to interpret genomic data from natural populations and experimental evolution and so on. So thanks again, Stephan, for telling us about your work. And I don't know, Patricia, if you want to say something.
Just to say thank you all and for the interesting discussion later. And thank you. And I will make it available for you to see again in the YouTube. Yes. Thank you.
Thank you.
Thanks.
Bye-bye.
Bye. Thank you.
Thanks, Stephan. I'll talk with you later.
Okay, bye.
Okay, bye-bye. Thanks.
Bye.